Joe has, I hope I'm not butchering your last name.
No, you got it.
Okay,
Yeah,
all right.
that's good.
Who has recently joined the Department of Psychology at CMU, formerly from Rochester. We're really happy to have him here in Pittsburgh. So Brad works actually on some topics I've been interested in for now almost 20 years. There is the nature of concepts and how they're represented in the mind and in the brain. And Brad has done some very important work in neuropsychology on the matter. And I think we're going to hear much about that today. So
Great.
The floor is yours. And for those of you who are psychologists and are not used to be coming to the center, you may ask questions, but the tradition in philosophy is to keep the question for the end. But if you have clarification question and if you can't really resist the urge, feel free to just interrupt Brad when he's going to be talking. Okay, the floor is yours.
Great, thank you. All right, well, thank you for the invitation to present today, and thank you for the introduction. Certainly, if people have questions, it's from my perspective, it's okay. But you know, when in Rome, so
Okay.
up to you guys. You can sort it out. Yeah. Okay. So I just came to Pittsburgh this past summer. So just started this past summer. So we're just really kind of figuring ourselves out as a lab and figuring out how to redeploy the research program that I'm going to tell you about, which was largely, most of the research I'm going to tell you about was conducted at the University of Rochester over the past 9 years. And then I'll maybe get to some of the future directions that we're excited to pursue here, particularly in collaboration with neurosurgery, the Department of Neurological Surgery at UPMC. So the research I'm going to tell you about is about the neural representation of object concepts. This is a topic that I've been interested in ever since I've been interested in psychology. I started working on this actually as an undergraduate. I don't know if there's any undergraduates in the room. Certainly I worked on it as a graduate student. And so for me, this has always been sort of the topic that's closest to my heart and really is the main theme that runs throughout the research in our lab. So for me, the starting point in thinking about the organization of object concepts in the brain is the idea, or I would even say the fact, that the human brain exhibits functional specialization. And by functional specialization, I mean that anatomically separable regions of the brain support functionally separable computations. Right? And understood in this way, this concept of functional specialization has a long and somewhat complex history. And it runs up through modern considerations of how the connectivity of the brain might shape functional specialization. And this is an idea that I'm going to come back to throughout the talk. And it's one of the main ideas that I'd like to suggest is that some of the kinds of functional specialization that we see in the brain for object concepts is driven not by local processes expressed where you see the functional specialization, but actually driven by the connectivity of those regions to other parts of the brain that are processing other types of information about the same category or class of items. But I take it as a fact that the brain exhibits functional specialization, an agreed upon fact or empirical consensus. And I think research has really shifted to understanding what are the factors or constraints that drive functional specialization in the first place. Are those constraints innate? Are they experiential? What's the relevant balance between these two extreme positions? Another set of questions that people are asking is, what is the granularity of functional specialization? Is the granularity of specialization really at the level of semantic categories or semantic domains? Or is there rather functional specialization for continuous dimensions that happen to statistically co-vary with semantic category in our experience? There's another aspect of functional specialization that I'm very interested in. I refer to it as the dark side of functional specialization. The dark side of functional specialization is borne out in the observation that some patients, after a focal brain injury, can have permanent neurological deficits. Now, the fact that, without even doing any experiments, the mere fact that a patient can have a focal brain injury and go on to have a lifelong cognitive impairment in a particular domain of cognition tells us, I think, two things. Number one, it tells us that it must be the case that cognitive domains or computations are somehow, to some degree, anatomically separable in the brain, such that damage could affect one process without affecting other processes. And the other implication, which really has to do with the dark side of functional specialization, is that it means that parts of the brain that were not damaged in that patient, that were not affected at all by the lesion, were not able to take over the functions that were compromised by the lesion, right? So the fact that you can have selective cognitive impairments means that the rest of the cognitive system was not, for some reason, was not able to adapt and take over the function that's been lost. Now, this isn't something, I don't think, that's particular to the brain. I mean, I think if one has the misfortune of, for instance, having their pancreas fail, it's not like the liver can just take over the function of the pancreas, okay? So there's a lot of functional specialization throughout biological systems, and I think, I would argue, the strong form of this is that the same principle really applies to the brain, and the consequence of that are the kinds of selective permanent injuries that you can observe in patients. So you might think of functional specialization and neuroplasticity as somewhat interacting or even dynamically opposed forces in the degree to which the adult system has achieved a certain degree of specialization and it has a certain rigidity associated with that organization. If you perturb the system's not necessarily going to be able to reorganize to take into account that injury. Another dimension that's very important in this mix is another concept is the concept of connectivity. And writ large, connectivity in the brain is what allows the computational outputs of one region to be the inputs of an anatomically distant region. But connectivity also has a dark side in the context of neurological injury that's referred to as diaskesis. Diaskesis is an old concept from neurology and philosophy, I think even before neurology, whereby injury to one particular part of the brain or a disruption to one particular process will have consequences for other parts of the brain and other processes that depend on that injured region. All right, so these four concepts, specialization of function, neuroplasticity, connectivity, and diaskesis, are really the four main concepts that structure my research program and structure what I'm going to tell you about today. I'm going to focus particularly on specialization of function and connectivity. At the end, I'll tell you a little bit about the work that we're doing to try to get at these issues of plasticity and diaskesis. But I wanted to try to go deep today as opposed to go broad, okay? So, but I just want to say kind of at the beginning, one could apply this kind of a conceptual framework to really any domain of cognition. In my lab, we look at this, we use this as the framework through which to think about object concepts. And in thinking about object concepts, one of the most important distinctions, I think, to start with is the distinction between a ventral visual pathway. And I apologize if this is review for some of you, but I want to make sure we're all on the same page. So the ventral visual pathway, just to orient people anatomically, this is the back of the brain, occipital cortex, primary visual cortex is back here. The ventral visual pathway projects from early visual cortex to regions in the anterior temporal lobe and supports object identification, probably in allocentric reference frames, okay, in spatial coordinates defined by the structure of the world as opposed to defined by our perspective on the world. And this is the main pathway for extracting object properties that are invariant to the particular way in which you look at a particular object in a particular situation, all right? So my perspective on that banana on the table over there is that I can't quite see, I can't quite see inside of the peel. Now I can infer that there's probably banana inside of that peel, but that's not something that's given to me perceptually. That's something that I have to sort of reconstruct based on what I know about bananas and what I'm perceiving there. And so the derivation of those kinds of invariant object properties requires, it's been argued, requires the kinds of processes that are implemented in the ventral visual pathway. All right, so in the context of objects, a particularly important type of information is the extraction of information about how to manipulate an object according to its function, and specifically what the function is. So objects, tools, all their manipulable objects have functions. These are the functions they were designed for. When I talk about object function, I'm not really talking about it as the design intent of the object, but as the goal of proximate use. So when you pick an object up in order to use it, you pick it up in order to satisfy some broader behavioral goal. It could be as simple as quenching your thirst to pick up a glass, or it could be something more complicated with a more complex tool. That higher level knowledge of what it is that you want to accomplish with the object is what I'm going to refer to as function or purpose of use. And arguably, that is derived through a ventral visual pathway analysis. Now, that's not a fact. That's a hypothesis that I'll come back to throughout the talk. One can contrast the ventral visual pathway with the dorsal visual pathway. All right, the dorsal visual pathway projects subcortically and cortically to regions of posterior parietal cortex and supports visual analysis in the service of object-directed action. So it supports visual analysis, for instance, in egocentric reference frames, and reference frames that are anchored with respect to the current location of the hand or the current location of the trunk or the head. And because ultimately that's the reference frame that's relevant for object-directed action, it doesn't matter where the object is in absolute coordinates with respect to other objects. What matters is where is it with respect to the starting position of the hand. So the dorsal visual pathway extracts information about objects, basic volumetric information about objects, spatial location, I'm going to argue orientation, and projects to posterior parietal cortex and supports online visual motor actions. And what I mean by that is that you have visual information coming into the system, and that visual information is used in real time, for instance, to guide the hand to an object, or in real time to guide the eye to make a saccade to a location in the visual field. So it's that real-time use of visual information in the service of action that the dorsal stream is particularly, has a sort of particularly privileged role in the system. Now, a lot of what we know about this kind of a parcellation comes from studies of patients with focal neurological injuries. So I'm going to give you some examples throughout the talk of the kinds of patients, the kinds of patient evidence that motivate this kind of a framework. One of the more famous patients that's been studied is a patient that goes by the initials DF. And patient DF was described by Mel Goodale and colleagues in a now classic famous paper in 1991 published in Nature. And patient DF had bilateral lesions to LO. So you can't quite see it in this picture, but essentially this over here and this over here, or this in the left hemisphere and its complement in the right hemisphere, were lesioned in this patient. And the lesion was caused by an anoxic injury from carbon monoxide poisoning. And this particular part of the brain is what's called a watershed area. It lies between two major artery systems, and regions that lay sort of in between two major artery systems are particularly susceptible to anoxic injury. That's the first bit of tissue to die. And so she had, unfortunately, these bilateral lesions in this watershed area that happened to be functionally the part of the brain that supports the extraction of object shape from visual input. So she has a very dense visual agnosia, and I'll show you some evidence in a moment. But what's startling about patient DF is that her visual motor abilities, based on real-time use of visual information, are essentially intact. So here is this patient playing catch, for instance, and you wouldn't know, watching her play catch, that she is densely visual form, has a dense visual form agnosia. Now, Goodale and colleagues studied her formally in a few different ways, and one of the ways in which they studied her in the initial paper was they gave her a card. I don't know if you can see it. The contrast is a little low. She's holding a card right here, and there's a slot, and her task is to post that card in the slot. And watch when she is asked to post the card in the slot, she doesn't really have any difficulty. She just reaches out and she puts the card in the slot. But if you change the task and you say, now, don't post the card in the slot, just adjust the orientation of the card to match the orientation of the slot. So now it's a perceptual matching task. Now you can see that she's saying the slot goes this way. In fact, the slot goes that way. So if you do that a lot and you test her across many trials where you keep varying the angle of orientation of the target slot and quantify her performance, in the matching task, and this is performance normalized to a given reference point, controls have no problem orienting the card to match the orientation of the slot. It's a pretty trivial task. Patient DF is really kind of all over the place, so she can't orient the card reliably to match the orientation of the slot. More simply, if you show her, if you hold up a pencil and you say, is this vertical or horizontal, she's at chance. She can't tell you about the structure of the world based on visual input. But if you ask her to post it, her performance looks much more like controls. Okay, so she's way better than she is actually for the perceptual matching task. And there's been other patients like her that have been reported, and there's also been, I'm not going to really get into it, so I'll just tell you, the reverse dissociation has been reported as well. Patients who have difficulty with visual motor transformations, but don't have difficulty with these kinds of perceptual matching tasks. So from patients like patient DF and other patients like her, one can conclude that the ability to perform certain kinds of visual motor actions is computed independently of whatever processes are supporting phenomenological perception, right, or seeing. But it can't be that simple. And I think even before getting into any experiments, if you think about the everyday situation, imagine that, imagine you're a righty like myself. Okay, so you pick up a pen or a pencil with your right hand to use it. Imagine you're holding a hot cup of coffee with your left hand. And now I need to pick up this pencil to write down a number. What is the grasp that I'm going to use to pick up this pencil, right? I'm going to use some kind of overhand grasp like this, which is quite biomechanically uncomfortable, but what it does is it puts the object in my hand in the right orientation in order to use it. We do this all the time. Compare it, if you're going to pick it up to use the eraser, you'd probably pick it up something like this, right? And we do this all the time. If you kind of just casually watch yourself interact with objects, you'll see that the way in which you initially grasp an object anticipates what you're actually going to do with the object once you have it in your hand. And this is a phenomenon that David Rosenbaum has referred to as end-state comfort, which is that we grasp objects in a manner to, generally speaking, to maximize the end-state comfort, to maximize the affordance structure of the object in your hand in order to actually then manipulate the object according to its function. Well, given that that's the case, and given that I just said that the dorsal stream is what actually supports the object-directed grasp, and the ventral stream is what supports the extraction of these kinds of invariant properties, well, then we have praxis. We have what it is that you do with the object. How do you physically manipulate the object once it's in your hand? What is the complex manipulation that's applied to the object to accomplish its function? And the inference that I would like to draw is that object manipulation knowledge is accessed via eventual visual pathway analysis, arguably because the kinds of praxis representations that we have about objects is not information that's given bottom up by the object, right? So when you look at a tool, and a tool is a particular way of manipulating it in order to accomplish its function, that's not transparently given just by the structure of the object. That's something that you also have to learn and have represented. And to access that kind of information, arguably, then you need to go through eventual visual pathway analysis. But if that's the case, end-state comfort tells us that the grasp that we deploy to objects take into account or are constrained by what it is ultimately that we're going to do with the object once it's in our hand. And what that suggests is that when we grasp objects in a functionally appropriate way, so in a way that anticipates how we'll actually manipulate the object, that grasp has to wait on information that's coming from the ventral visual pathway in order to constrain the kind of initial grasp that's going to be appropriate to that object. All right. That's essentially a hypothesis, right? That's not a, I don't mean to present that as a fact, but what I'd like to do is kind of take you through some of the reasoning throughout the talk that leads me to think that that might be the way that it's working. Now, one thing may have occurred to you, which is that if, on this kind of an analysis, and I should say that by these arrows, I don't mean processing direction. I mean the kinds of constraints that are carried forth from one separable process to another separable process, right? So grasping is constrained by what you do with the object when it's in your hand, but what you do with the object when it's in your hand is constrained by what it is that you're trying to do with the object in the first place, but you can't get that without recognizing the object in this allocentric reference frame, right? Patient DF had bilateral ventral stream lesions, and she had no access to this information. So the expectation would be that she would not show the phenomenon of end-state comfort. She would not grasp objects in a manner that anticipates how to manipulate the object once it's in her hand. And this is actually the case. So for instance, patient DF was tested for grasping objects. And if you orient the object like this, such that the handle of the object is pointed toward the subject, such that there's essentially a confound between the biomechanically comfortable way of grasping the object and the functionally appropriate way of grasping the object, then DF appears to maximize end-state comfort, as do healthy controls. But if you flip the object around So now you have to use a biomechanically uncomfortable or non-canonical grasp in order to respect end state comfort. Controls have no problem doing this, but patient DF will continue to grasp the object in essentially a volumetrically and biomechanically appropriate manner, but in a manner that doesn't actually facilitate end state comfort. So she might grasp the screwdriver like this, right? And then once it's in her hand, she can recognize the object based on tactile cues. She just can't recognize it based on visual cues. She can recognize it based on tactile cues, and then she can reorient it, and she doesn't have difficulty with manipulating objects, so she can then use the object. Okay, so this is a framework for thinking about some of the separable processes that are involved in object recognition and object use. And I would be the first to admit that this is a cartoon, and let me just expand a couple of the boxes to begin with. So for instance, I've co-located in one box identifying an object. Well, that's an incredibly complex and dissociable set of processes in and of itself. Just to give you a sense, I think one can usefully distinguish between object identification based on visual form and based on surface texture properties, right? So, for instance, patient DF, who I was telling you about, she has difficulty recognizing objects based on visual form, but she has no problem appreciating, for instance, recognizing the color of objects, recognizing whether something is slippery or rough or soft or hard. She doesn't have difficulty with analysis of surface texture properties. She just has a problem with visual form. And the reverse dissociation has been reported as well. Patients who have lesions, and we'll get into the regions in a moment, but patients who have lesions to a separate part of the temporal lobe can have difficulty appreciating surface texture properties about objects, but not have difficulties recognizing objects based on their visual form characteristics. Yes?
Can we recognize form if they're texture defined?
How do you mean? Like, I think I know where you're going.
Differential textures across an object, which indicates some kind of shape. Do that.
Yeah, so for instance, if there's really strong texture cues,
yeah.
So for instance, like, okay, people are eating, they're eating fruit, and then I see a yellow long, a yellow piece of fruit on the table, she would certainly be above chance calling that a banana, right? Exactly for those kinds of cues. But now if that was black and white, for instance, you took away those texture cues, then all of a sudden her performance would fall apart. So yeah, so she's very good at, and these patients patients generally are very good at using the things that they still have functioning well. So she'll probably rely even more so than one typically would on those kinds of cues. Yeah. This is an example of a patient that we studied in our lab who had a lesion this is the temporal lobe here, shown in a coronal section, and you can distinguish the ventral surface of the temporal lobe, which abuts the cerebellum, and the lateral surface of the temporal lobe. Patient DF had lesions on the lateral surface, and lateral posterior lesions typically lead to impairments for visual form. Ventral lesions on the ventral surface can lead to impairments for surface texture. All right, and this will actually become important later in the context of the fMRI data that we'll talk about. Well, this is a stroke patient. This patient was a particularly interesting patient because he had no difficulty naming colors, recognizing colors. His color perception was completely intact. But if you asked him what color is the sky, what color is a banana, and he couldn't look at one of those stimuli, then he had absolutely no idea. So we did a bunch of tests with this guy. But for instance, you give him a box of crayons, go through, name the color of all the crayons, no problem. Name some pictures. You can name the pictures. Now take the crayons and color in the pictures according to their appropriate colors, and then he just comes up with all kinds of weird assignments of colors to objects. Okay, there's just some examples, right? So green carrots and that kind of stuff. And this is a way of quantifying patient performance that I'll use throughout the talk. So this is patient performance normalized to controls, expressed as t-values, t-scores. So you can think of these units as standard deviations. So the patient's color knowledge is, like 5 standard deviations below control performance, but knowledge of visual form, knowledge of biological motion properties, those are around 0 or slightly positive, meaning in the control range. Okay, so one can distinguish these, you can kind of break up object identification into these two sub-processes. And I'd also like to argue that the dorsal visual pathway has its own appreciation of the visual structure of objects quite independent of the visual pathway. And there's actually a fair amount of evidence that's come out in the last several years, also from Marlene Berman's group in the Department of Psychology. I want to show you some patient evidence that we've recently published. And the patient I'm going to show you had a lesion to primary visual cortex. So this is just a little primer on the anatomy. This is primary visual cortex, and 90% of the ganglion cells in the retina send an axon that goes to LGN and then on to primary visual cortex. It is 17, yeah. Well, 17 includes primary visual cortex, and it includes V1, probably V2 as well. About 10% of the ganglion cells in the retina send axons that go to superior colliculus and pulvinar and then directly onto higher order visual areas, or to LGN and then directly onto higher order visual areas, essentially bypassing V1. And what this means is that you have pathways of visual input to the brain that bypass primary visual cortex. Okay, so now we have a patient who has a lesion that's affecting primary visual cortex. This is the patient's acute diffusion weighted image. And this stroke lesion, actually what it did is it deafferented primary visual cortex. This is primary visual cortex. This stroke lesion blocked the what are called the optic radiations going into primary visual cortex. This is a representation of the patient's phenomenal vision as measured through a standard clinical test of visual abilities. Black on this scale means blind, and white means vision is intact. So this patient had what's called the lower right quadrantinopia. He's blind in the lower right quadrant. And so we tested the patient in this manner. So the patient's going to be sitting at a table. He's going to grasp a handle, and this handle can rotate 360 degrees. If he fixates over here, this handle falls in his blind visual quadrant. If he turns his head and fixates over here, the same handle now falls in his intact visual quadrant. And what we're interested in asking is whether or not he can orient his wrist in flight prior to making contact with the handle. Can he anticipate the orientation of the handle based on visual information before he's actually made contact with it when the handle's presented in his blind visual field? So here's a video of the patient doing the task. He's grasping the handle in his blind field. And just watch his wrist just prior to making contact. So you can see there the handle's horizontal, his wrist is horizontal. Now the handle's going to be vertical, and watch his wrist. So he's got his wrist vertical, all right? And so this is how we tested the patient. We tested this patient as an inpatient with this kind of a setup. Here's the same video sped up so you can kind of appreciate it with freeze frame. Now he's wearing a iPod with an accelerometer app on it, and that's how we're actually going to measure his wrist orientation in order to quantify whether or not he's actually anticipating the orientation of the handle. So if you plot the handle orientation against his wrist orientation, you can see that there's some sensitivity in the sense that as the handle is changing its orientation, his wrist is changing orientation prior to making contact. And what this tells us is minimally is that these non-geniculate striate pathways, these pathways that I was saying that bypass V1 and don't, well, presumably they don't contribute to conscious vision because he's blind in the area of his visual field for which these pathways are intact, but V1 is not intact. He's still able to change the orientation of his wrist. Now, if you change the task, if you recall from patient DF, they tested DF both with a grasping or with a pointing task and with a perceptual matching task. So if you change the task now and you say, don't grasp the handle, adjust the orientation of a second handle that you can see, that's in your good visual field, to match the orientation of the handle that you cannot see, then his performance falls apart. And this is essentially, internal confirmation within the same experiment that in fact he's blind in that region of his visual field. And if you test both of these conditions in his intact visual field, of course he's fine with both of them. Okay. All right. So what those kinds of data, I find those very compelling in that they demonstrate that the dorsal visual pathway seems to have information, rudimentary information perhaps, but have information about visual structure, basic volumetric properties, and probably orientation information about objects. All right, so if you put it all together and color code it in a way that we can then map it onto the brain, this is sort of the full framework. So the data that I'm showing you plotted in the brain, these are actually real data from about 40 healthy participants. and they were in an experiment in which they were looking at or naming pictures of common manipulable objects or various baseline categories like animals. And if you say what parts of the brain are more activated for naming tools than naming animals, these are the regions that you get. With the exception of LO, this is a region that was lesioned in that patient DF bilaterally. You define this region actually by all objects versus scrambled images. But the point, and so what I've done is I've color-coded the neural data according to the color coding down here. The parcellation of these boxes is really based on the patient evidence. And this kind of shows you where lesions create these kinds of selective deficits in patients, all right? So for me, this is really the starting point. This is, I mean, it was like a concerted effort to kind of get to this point, but really now we have, I feel like, a framework within which to start to ask questions about the neural system that supports object representation. Let me just put in a caveat now as opposed to at the end of the talk, which is that I do want to say that while we do a lot of functional MRI in the lab, and it's one of our main tools, I think a healthy dose of skepticism is warranted about what these kinds of data are actually telling us about how the system works, right? If we care about how the system works, then we should think carefully about what maps are actually telling us about how the system works. And I'd suggest that a map's relevance to an explanation of behavior is only as good as the labels, right? What it's a map of. And I didn't update my image here, unfortunately. This is from when I gave a talk in Boston instead of having a picture of Pittsburgh. But you know, if I was someplace in Boston and I had to get to some other place in Boston, and I said to somebody, "Hey, can you tell me how to get to Harvard Square from where I am right now?" And someone said, "Oh yeah, here you go. Here's a satellite image of Boston. Now you should be able to find your way." So I can't figure out where I'm going just from this image, right? And well, because I don't know where I am in this map, and I don't know where I need to go in this map. Now, if you added some labels to the map, it would start to get a little easier. Now, the way in which we normally interact with maps is like this, right? We just abstract out, and essentially we're just looking at spatially organized labels, right? Now, if I look at this kind of a map and I figure out how it is that I'm going to navigate from my hotel to where I need to get to, and I figure out what the directions are, and then I take away the map, arguably my ability to navigate to where I need to get to is not negatively affected by taking away the map, right? So the point is that the magic of the map is in the labels, not in the picture. And this is probably a point that is almost pedantic in this group. It's not pedantic in many empirical circles, but I do think it's sort of worth keeping in mind. And this is something that's been thought about a fair amount. David Popple, somebody who's written about this, called it the mapping problem. All right. So if you take that the outcome of that back to what I was talking about, the relevance of this map is really dependent on the parcellation of the processes that we're assigning to different regions, all right? And that map is really only as good as our ability to parse the system in this sense. All right. So let me go through a few things. I want to make sure that I leave enough time for questions. So let me tell you about, I've told you about several different kinds of patients. One class of patients that we've been particularly interested in studying are patients who have difficulties with manipulating objects correctly according to their function. This is a video of a patient. I spent 3 years in Italy in graduate school, and this is a video of a patient in Italy. And you'll see that he has some quite dramatic difficulties for manipulating objects properly according to their function. So here he's given a pair of scissors. There's a very strong cue. There's a piece of paper there on the table. He doesn't have any obvious loss of dexterity or motor control or visual motor coordination. He can pick up the scissors, he can manipulate them, but he really is sort of at a loss for how to manipulate this object according to its function. Now, what's interesting about these kinds of patients is that they can tell you all kinds of things about the object that they can't manipulate. So sometimes they can tell you its name, they can tell you, where you might find it, they can tell you all kinds of sort of encyclopedic or conceptual knowledge about the object that they don't seem to be able to physically manipulate. And this is something that's been studied. There's maybe 15 or 20 well-described cases like this in the literature that demonstrate this dissociation between impaired praxis, impaired object manipulation, and spared conceptual knowledge. So, you know, like I said, you know, this was done in Italy, and IRB is a little lax. So, you know, let's give an apraxic patient a gun, right? But you notice, like, the way he's picking it up, he'll actually kind of get it, he gets the affordance structure backwards. He puts his, but, you know, you would never hold a gun like that, right? Even if you were 99% sure that it was empty, you wouldn't hold it pointing toward yourself, right? Playing around with the trigger hold. All right, so compare that patient to this patient. Here's another patient. This is a patient that we studied in great detail in the lab. This is his demonstration of how to use scissors. He's actually much better, right? He's not correct, but he's much better. He's got a lot more correct about the affordance structure of the object, but what he's messing up are what are called spatial and temporal properties about the object. Whereas this patient was making errors of content, and that's a distinction that's made in the apraxia literature among the different kinds of errors that these patients can make. This corresponds for any clinically minded folks between ideational apraxia and ideomotor apraxia. Okay, so if we stick with this patient, this is the patient's performance on that same scale that I explained for spatial errors and temporal errors for using objects. This is the patient's naming ability for the same tools that he can't use. All right, so this is just another demonstration that you can have impaired use and spared naming. And if you take object naming to be an index of the integrity of lexical, semantic, or conceptual information, then you can infer that you can have impaired use and spared concepts. This is a larger case series that I worked on in graduate school that was carried out in the lab of Raffaele Rumiati in Trieste in Italy, of a series of unilateral stroke patients who were all tested on object use and object naming. And you can see that across the group there is, you know, a very clear double dissociation represented. Some patients are impaired for naming, but okay for use. Some patients are impaired for use and okay for naming. Okay. Now, some of you may have encountered a framework, theoretical framework about the representation of object concepts that's generally referred to as embodied cognition. I know you've written on this topic a fair amount. For me, I'm not going to get into talking about embodiment that much today because mostly it's a negative argument coming from me. But I do think that these kinds of neuropsychological data are essentially devastating for any strong form of the embodiment perspective. If you have a theory of concept representation that says that the content of concepts is in some interesting sense represented in the sensory motor systems, and then you have patients who have lesions to the sensory motor systems and frank impairments for sensory motor abilities or processes or content, but they don't have conceptual impairments, then in my mind, the theory needs significant attention, okay? We'll come back to that a little bit. So let me tell you a little bit about some fMRI that we've done to try to get at these issues. And one of the things that we were particularly interested in looking at was, let me just back up here a little bit. So, you know, on the one hand, I just told you, you can have an impairment for object manipulation that spares conceptual knowledge about the same objects you can't manipulate. At the same time, I showed you these data, and I said that we obtained these data in healthy subjects when they were naming tools or naming animals. So there's a bit of a paradox there, because on the one hand, you have activity of a region during a naming task that the patient data tells you don't need to have that region in order to perform the naming task. So the question is, what is represented in this region? Question one. Question two is, why is it activated? So I'm going to show you an experiment that addresses the first question, or moves the needle a little bit on what we think is represented. And then the second question we can come back to in discussion. So how do people here, show of hands if you know what multivariate pattern analysis is in fMRI? Okay, a few people. So let me just give a little bit of background on this, and I'm going to try to not strand my ship on the rocks of Ven's recent publication. Okay, so so what we did is we came up with a stimulus set, for instance, bottle opener and corkscrew. Now, bottle opener and corkscrew are similar in terms of their functions. They're both used for opening bottles. And now we can add a third item to that triad. So we have screwdriver. Now, screwdriver is similar to corkscrew in terms of the way in which you physically manipulate the object, but it shares none of the function. So we have these kinds of triads where you have two items are related in action and two items are related in function. And here's another example of a triad. So knife and scissors are both used for cutting. Scissors and pliers are both used with a similar manipulation, but have very different functions. Now, if you show people images of these objects and have them really do any kind of task over these, you're going to get about the same amount of activity is going to be elicited in the parietal regions that support object use. Right? So all of this network of brain regions that I showed you that support tool knowledge, they're all going to be activated more or less the same in terms of the amplitude of the response for pliers and for scissors and for knife. But there's another way to think about analyzing the data, which is to extract. So this is, a functional MRI data set consists of, at its most fine-grained resolution, spatial units that are called voxels. So you can imagine taking the entire Euclidean space inside of which is the brain, and you break it up into little voxels that are 3 millimeters on a side. Three little cubes that are 3 millimeters on an edge. And there might be 50,000 of these little cubes that make up the brain. Voxels are the 3D analog to pixels in a photograph. So it's the smallest, it's the highest resolution, spatial resolution. So if you take a region, for instance, parietal cortex, and I said this region of parietal cortex responds the same amount to pliers and to scissors and to knife and to screwdriver, etc. If you extract the data from this region, and these are, say, the voxels that fit inside of this region, and you take all of the response profiles of each of these voxels, and you transpose it just into the form of a vector here. So this is, for instance, all of the voxels in parietal cortex and how much each of those voxels responded when the person looked at corkscrew, and how much each of those same voxels responded when they looked at screwdriver, when they looked at bottle opener, et cetera, right? Multivoxel pattern analysis, multivariate techniques, multivoxel pattern analysis is based on the idea that there is information contained in the pattern of responses across this population of voxels. And there's many ways of capturing that kind of a pattern. And if you want to know the details of the more advanced methods, then would be a great person to talk to. In the experiment that in the study that I'm going to show you, we used a kind of a stock machine learning algorithm called support vector machine classifier, linear classifier. And what we did was we trained a classifier to learn what is it about the patterns of the voxel patterns in parietal cortex that distinguishes scissors from corkscrew. Right? So this classifier's job is to learn what is it about these patterns that's different for scissors and for corkscrew. Then you take the classifier that's been trained to learn the distinction between these two different objects, and now we're going to test the classifier on a separate set of objects. And that's kind of the main innovation of this study, is we train the classifier on scissors versus corkscrew, then we test the classifier on pliers versus screwdriver. So if the classifier has learned something about what distinguishes scissors from corkscrew, and it's learned something about it, its ability to distinguish these objects is based on it having learned something about how they differ in their manner of manipulation, then this classifier should also, the same classifier that's been trained on these objects should now be able to be transferred to decode pliers versus screwdriver, because pliers are similar to scissors on manipulation, and screwdriver is similar to corkscrew on manipulation, all right? So this is a way to, what we call this cross-object decoding. You train on one pair of objects, you test on another pair of objects. Successful transfer above chance accuracy of a classifier means that it's learned something about what distinguishes the objects. In this case, it's manipulation. Now, the nice thing is we can take the same classifier that's trained on scissors versus corkscrew, and we can test it on knife versus bottle opener. And now if the classifier transfers, it means it's learned something about the function of these objects that is allowing it to now distinguish between this new pair of objects. So what we did is we had these kinds of stimuli, screwdriver, corkscrew, bottle opener, and participants performed two different tasks. They were either shown printed words, like the word screwdriver, and when they got the word screwdriver, they had to, during the scan, they had to actually pantomime how you would manipulate the object according to its function. In other runs, the participants performed a very difficult perceptual and back task where they had to recognize repeat exemplars from the same basic level category with overt pictures. So in one task, they had printed words and they had to produce an overt pantomime. In the other task, they saw pictures and they had to perform an identification task. And we can decode manipulation similarity, or we can decode function similarity, as I just described. And I'm going to show you results from whole brain analyses and leave out the ROI analyses for now. So what we find is that regions that are able to decode similarity and function are largely restricted to the ventral visual pathway. And that's the case both for the pantomime task and the identification task. Regions that can decode manipulation similarity during the pantomime task include pretty much all of motor and somatosensory cortex. And for the identification task, a much smaller subset, including this region of inferior parietal cortex. All right. So this is already quite a sparse representation, or a subset of the regions that are able to decode manipulation knowledge independent of task, or function knowledge independent of task. But what we can also do with this experiment is we can do cross-task decoding. And I hope that people are still following. If there's clarification questions, please ask me. We can train the classifier on pantomime data and test it on pantomime data. And that's what I showed you. Or we can train it on identification data and test it on identification data. And that's what I showed you. Or we can train it on pantomime data and test it on identification, or vice versa. And the value of this approach is that it tells us whether or not there are multivoxel patterns that are invariant to task, right? That they're there regardless of whether you're pantomiming or whether or not you're identifying the object. And what's interesting is that the region that falls out of this analysis is always this inferior parietal. So the only region of overlap here is essentially that inferior parietal supramarginal region that I was talking about earlier, which is color-coded yellow in this image. Okay, so what I take these data to suggest is that neural activity in the left inferior parietal lobe when processing tools indexes compulsory access to abstract representations of specific object-directed actions. They're abstract because they're invariant to task. It's compulsory, presumably, because we know that you get activity in this region in a naming task, but if you have a lesion to that region, you don't have a problem with naming. So it's being activated even though it really doesn't have to be activated in order to complete the task. Okay, let me just keep going a little bit. In the interest of time, I have 2 minutes. I have a little bit more content that'll fit in 2 minutes, but maybe I'll take 7 if that's okay.
That's good. Yeah.
Okay. I want to tell you about one more study. So one of the questions that I've been particularly interested in, and it's sort of a hypothesis that we've been that's structured a lot of our current research, is asking, why is it the case, sorry it's a little dull, but why is it the case that you have regions in the visual system that exhibit neural specificity for small manipulable objects? Small manipulable objects are defined, largely defined compared to other categories in terms of what we do with them, in terms of motor properties associated with these objects. So why would you have neural specificity in the visual system for a class of items in the world that are defined by motor properties, defined by properties that are expressed by another region of the brain? And the hypothesis that we've been looking at is that the reason why you have the emergence of what looks like neural specificity here for tools is really the conjunction of two constraints. One constraint is coming from regions in parietal cortex that support object-directed grasping. And I apologize, let me just go back instead of going forward. Yeah, here's a good one. So this region in red here supports hand shaping in order to grasp objects. Lesions to this region cause deficits in proper hand shaping in order to grasp objects. These regions in the ventral stream, there's prior evidence. I showed you a little bit of evidence from patients showing that this region cares about surface texture properties of objects. So the idea is that when you are going to grasp an object, you have to take into account information about its volume, about its location, its orientation. You also have to take into account information about its surface properties. Think about the way in which you grasp a slippery wine glass when you're doing the dishes versus how you grasp it at a dinner party, right? It's very different, right? You have a really sort of maniacal grasp on it when you're doing the dishes so you don't drop it. But in a dinner party, of course, you're much more dainty. And so so the idea is that when you grasp an object, you need to know about, for instance, the coefficient of friction of the object. You need to know about the weight distribution in that object in order to be able to grasp the object in the appropriate location. Well, if this is the system that's actually supporting hand shaping and targeting of the hand to objects, and this is the system that's supporting surface texture analysis, then the hypothesis is that this system is essentially querying the ventral stream and saying, tell me about what you know about the surface texture properties of this object. But it's only doing that for manipulable objects. It's not doing that for animals and other things that we don't typically grasp, right? So if you recall, I showed you data from a blind side of a patient who had a lesion to V1. The patient was still able to grasp the handle in the blind field. What those data tell me is that the dorsal visual pathway is able to deploy volumetrically appropriate grasps to objects independently of analysis in the ventral visual pathway, very much like patient DF, which would suggest that this system is able to deploy a grasp to an object even though it doesn't know what it's grasping. And it doesn't know anything about its coefficient of friction or its weight distribution, those kinds of things. That information has to come from the ventral visual pathway. And the constraint that's expressed by querying that information only for graspable objects is what creates what seems to look like actually neural specificity for tools in the ventral visual pathway. All right, in the interest of wrapping up, let me just say the strongest prediction that I can imagine being made by that theory is that if that's the case, if you have a lesion here, it should disrupt your tool responses in the ventral visual pathway, right? If you take this system offline, then you should see reverberations of that damage, also diaskesis, okay, in the ventral visual pathway. So that's the study I want to show you. Okay, so what we did is we studied 35 patients who had brain tumors, and all of the patients and a group of healthy controls named animals, faces, places, tools, and scrambled images. And what we did is we then, in the ventral stream, we extracted the amplitude of tool preferences for each patient. So in the temporal lobe, we extract how strongly does their temporal lobe respond to tools. I should say none of the patients actually had tumors in the temporal lobe. Their tumors were in other parts of the brain. And what we wanted to know then was whether or not the amplitude of tool preferences in the ventral stream is modulated according to whether or not there's a lesion in parietal cortex, in AIPS. Okay. So these are the results of a whole brain analysis. And what this analysis did is it took tool responses in the ventral visual pathway and correlated them with lesion location throughout the brain. Red, orange colors here mean that if there's a lesion, the likelihood of having a lesion is inversely related to the amplitude of tool preferences in the ventral stream, which is to say, if you have a lesion here, you have lower tool preferences in the ventral stream. All right, so this is the kind of evidence that you'd want to see if it's the case that tool preferences in the ventral stream are contingent to some extent on inputs that are coming from AIPS. If you take out AIPS, parietal cortex, then you wind up having lower tool responses in the ventral visual pathway. All right, we did controls. We did other analyses. I'm going to skip that just to skip through. Maybe I want to save time for questions. Let me just show you one other. I apologize. It's a bit of a schizophrenic talk, but I want to at least give you some thoughts and then tell you a little bit about what we're doing in the OR that we've been doing the last 3 or 4 years, and we'll be building up here at UPMC. So we study stroke patients. We also study brain tumor patients. We study brain tumor patients using functional MRI, and this is an example of a patient who had a tumor in his parietal lobe. And these are the fMRI experiments that we did in order to localize functions. And we share all this, we work with the surgeons to give them this information prior to surgery so that they can try to deduce the best way to get into and extract one of these tumors without causing deficits for the functions that are arrayed around the tumor. We also do diffusion tensor imaging to map major white matter pathways that are adjacent to tumors. In this case, this tumor has actually infiltrated this major white matter pathway. And we also follow patients into the operating room. This is what it looks like at the very start of the surgery. So this patient has been set in pins, is under general anesthesia, and we set all this equipment up, which allows us to present stimuli to the patient, record responses with a microphone, and have a good video recording of the patient's behavior. Now, once the surgery starts, all of this equipment is under the drapes. And the way the surgery happens is the patient is under general anesthesia for being set up, for the incision, for the craniotomy, for removing the bone flap, and for opening the dura. And then local anesthetic is applied around the site of incision. The patient is woken up in the operating room, and they can be comfortable and pain-free. And now they perform cognitive testing, while the surgeon has a electrical stimulator with a positive and negative electrode tips that is touched to the surface of the brain. And when this electrode is touched to the surface of the brain tissue completes the circuit between the two electrodes and shuts down processing local to the site of stimulation. If the patient's performance in the task suffers as a function of stimulation, then the surgeons know that this particular region they just stimulated is actually critical for that task, and then they would try to operate around it. And so what we do is we have been working to turn this into a research opportunity and also synergistically try to provide additional information to surgeons about functional localization. So for instance, we put this little registration star on the bipolar stimulator, and then through equipment that's in the operating room, we can register all of the points of intraoperative electrical stimulation in the same space of all of the preoperative MRI data that we've already collected in these patients. And show you a little bit of what this looks like. Is this working? You hear that? Sorry. Okay, here we go. So I'll play this again. So this is the patient. She's having surgery on her left temporal lobe. She's laying on her right side. This is what she sees. Her task is to say that is a fence. Okay. This shows you where the stimulation here is actually being delivered on her own anatomy, and then watch her performance here. So that's a phonological error.
This is a half mouth.
Okay, that's a phonological error.
Here is a vowel.
Should be a wreath.
Here is a tree.
Should be a deer. Okay, so in this case, stimulation of the superior temporal gyrus caused phonological errors, but watch what happens when she's stimulated here.
Watermelon. Here is a cat.
So you can see she has what's called speech arrest.
This is a key. This is a unicorn.
So when she's being stimulated there, she can't talk, and then as soon as the stimulation comes off, she can continue on with the task. Okay. So we for me, I'll skip the details here, but what we're doing with this is it's a way of sort of bringing the cognitive neuroscience laboratory into the operating room, and it's an opportunity to collect data that allow causal inferences about the necessity of a given region for a given function without the patient having a permanent injury, right? So it's a reversible causal disruption of processing. And a lot of this research is in the domain of language processing, but we've also had the opportunity to map a number of other domains that are not traditionally mapped in the operating room. I'll show you some data from a case in which we mapped music, but also mathematics, motor function, various types of language processing. So we're trying to push the envelope in the kinds of cognitive testing that can be done intraoperatively. And then we're also on the back end, doing this systematically and building a data consortium so that we can bring all of the data from all of these different cases into a common space to be able to ask with a higher N, right, higher power. Ultimately, the idea would be with hundreds of patients, questions with causal evidence about the organization of these processes. So just to show you one other thing, more show and tell than science, but this is a case of a patient, a musician who had a tumor in his right temporal lobe. This is fMRI localization of music in this patient's brain. And so this patient was prepared to play his instrument during surgery. This is preoperative. This is him practicing what it'll be like to play his instrument given the ergonomics of the surgical setup. In the surgery itself, he performed this kind of a task where he had to hum back a melody. and I'll show you what a couple errors look like. So you'll see that when the patient gets stimulated, he makes an error in pitch on this trial for musicians. And he also would spontaneously report his experience was altered. Okay. So what we found is that when the same region that was shown with fMRI to be involved in music processing was stimulated, then he had difficulties with the task. And okay, so just to give you an example, I mean, that was technically demanding to do the saxophone in the operating room, but you know, I think it's an opportunity to push the envelope in terms of what's possible clinically, what kinds of functions can be mapped clinically in these kinds of situations. And then I think it's an amazing scientific opportunity, because the only time that you have this kind of access to the human brain is if you work in this kind of a clinical context. So it's slow and messy research that takes a very long time to come to fruition. But this is what I'm going to be working on for the next forever here. So, you know, I'd be happy to talk about it further, maybe in a different context. All right, so question time.
We'll take a 1 minute break in case some of you need to leave, and then we'll have 20 minutes for questions. But already raise your hand if you've got questions, and I'll make a list.